Neste ut er også en som var med oss i går. Andy Taylor fortalte oss om hvordan man kan bruke mykorrhizadannende sopp til å restaurere skogsområder der lynghei har overtatt. I dag skal vi i større grad se på helheten av soppsamfunnet i de skotske fjellområdene, med foredraget Scottish Mountain Fungal Communities. Welcome back, Andy.
Thank you very much. Good morning, everyone. Hope you all had a good night. So yesterday I spoke a wee bit about fungi on the west coast of Scotland, and today I'm going to take you up into the mountains. This picture some of you may recognise is not in fact Scotland, but it's a lovely picture of Svalbard. I went there a few years ago with Hobart and I had a fantastic time. So we're going to go to the Scottish mountains, and when we started looking at the fungi in Scotland and mountains, there was incredibly few records, and I wanted to first of all see what was there, and I started looking at the fungi that were associated with ectomycorrhizal hosts, because we do in fact have some ectomycorrhizal plant species that grow up in the mountains, which are not trees. This is a way above the tree line. And we looked at 4 different species. We wanted to look at the communities on the species. So we looked at Archistaphylos, both the... Oh, I thought it was there. Okay, that's a good start. Okay. So that was the picture from Svalbard. So, as I was saying, we started looking at the plant species that we get up in the mountains. And we looked at 4. So we have Archistaphylos uva ursi, bearberry, and then the alpine species, Alpinus, the Betula nana, which is the dwarf birch, which you guys have a lot of. This is me standing in the best Betula nana forest in Scotland. It comes up to just about my knees. It's a fantastic area, fairly close to Aberdeen, in fact. And then we have Salix herbacea, tiny little plant, maybe just a couple of centimeters high. And all of these guys support ectomycorrhizal fungi. And what we wanted to do was look at the communities associated with these plants, and we wanted to look at the drivers or the factors which affected the communities. And because they're such poorly known systems, we wanted to look at how the nitrogen deposition, pollution, affected the communities. But also in Scotland, we have this very strong east-west gradients of temperature and also precipitation. So we go from maybe 600 millimeters of rain on the east to over 3,500 millimeters on the west. So we have a very strong gradient over a fairly short distance, and it allows us to look at all sorts of things. So we looked at all the species that I told you, the plant species, and we looked at both above ground. So we looked at the fruit bodies, but we also went below ground and used molecular tools to identify the fungi on the roots. If you've not seen the ectomycorrhizal roots of, say, bearberry, they look just like the ectomycorrhizal tips that you get on pine or birch. These are just pictures of a few of them, and they're completely covered in the fungal mycelium. So we extracted the roots, extracted the DNA, and identified the fungi associated with them. So we collected, as I said, all the fruit bodies and ended up with an amazing collection. And we identified about 260 species and huge diversity, much more so than we were anticipating. And many of these will be familiar to you. It's a rather strange situation where you have very, very small host plants and then sometimes very large fruit bodies above them. So you get an understory of the plants and then an overstory of the fungi. And it's quite a new situation to see that. Some of these you'll recognize as being the specifics of more commonly encountered ectomycorrhizal plant species. So in the middle there, you've got Swillus variegatus, which we consider to be specific on pine. And then bottom bottom left there, you've got Lixinum, which is quite often specific on birch. So these fungi are growing in association with plants which you don't normally associate them with. And in this case, it's usually Arctostaphylos, because it picks up all sorts of ectomycorrhizal species. Some of them are very typical of the habitat. Cortinarius alpinus, for example, we find it commonly associated with Salix herbacea. And you can see the very small oval leaves of the Salix herbacea underneath the fruit bodies here. We had some surprises, as you might suspect. We found Cortinarius saginus here, which on continental Europe at least, it's considered to be a spruce specific. But we found it with Betulinana, so dwarf birch, which was a real surprise to us. And I'm still trying to work out how or why that switch in host has occurred. We don't usually get saginus even with spruce in the UK. So this is a bit of an anomaly for us. So lots of species, including lots of these little brown guys, Cortinarius, all the Telemonias. Many of them are without name, I would say. So like I said, we had about 260 taxa identified. About 80 of them appear to be restricted to the alpine habitat. We had lots of new records, not surprisingly. So 36 new records for Scotland, about 28 for UK as a whole. But what was perhaps more surprising is that we consider maybe 23 of them to be undescribed and potentially new species. And if you consider that we've only looked at about 30 sites and we found 23 taxa that might be new, what's the rest of the diversity like? And can you imagine finding one new bird species, for example? The interest in that would be enormous. But we found all these new fungi and people went, oh, okay. So it was a real reflection of the interest in different parts of our biodiversity. So that was the first piece of work which I presented. Is really it's quite a few years ago now that we've done this, and the focus was really primarily on ectomycorrhizal fungi associated with particular plant species. But what about all the rest of the fungi which are down there? And I've teamed up with a colleague on the right here, Andrea Britton, who's a real mountain woman. And we've done a lot of work over the last 5 or 6 years looking at fungal communities, plant communities, and the interactions between above and below ground. And much of what I'm going to say now has been done in conjunction with Andrea. And I'll show you a few studies that which we've done or are ongoing. So one of the habitats which will be familiar to, I'm quite sure, is the Racemytrium moss heath. And it's in the UK, at least, it's really an iconic habitat. And it's very important for a number of reasons, including being the nesting site for some rare bird species, including this guy here, in English, the dockerel. And the dockerel has declined enormously over the last 10 or 15 years in the UK. And it's one of the potential reasons is the loss of good Racemytrium moss heath. Because what's happening is the pollution in the UK is affecting the moss and it's growing faster, but the decomposition of the necromass, or the dead material, is actually faster than the moss can grow. And the mats are becoming thinner, they're becoming fragmented, and other species are coming in and taking over from this particular moss. So we did a study down the UK, you can see on the left-hand side there, from way up in the northwest of Scotland, where things are still very nice, down to southern Scotland, into the Lake District, and then down into Wales, where things are really degraded, seriously degraded. We struggled, in fact, to find good moss mats in which to do our studies. So what we did was to look at the fungi associated both with the moss itself and then with the soil underneath the moss. And you'll be surprised possibly, although there's been some great work done in Norway on the fungi and mosses, that mosses support a huge diversity of fungi, quite often as growing as endophytes within the leaf cells and each individual cell. And we've had up to about 60 species of moss within a single shoot of the moss. It's quite incredible. So huge diversity within the moss. And what we've actually found, rather than a decrease in species, quite often you get a negative response of fungi with nitrogen increases. We've actually found that species richness increases with increasing nitrogen, both in the moss shoots and in the underlying soil. And it would appear that the nitrogen deposition is actually alleviating or removing nitrogen limitation on the moss communities, and they're proliferating. And this is one of the potential reasons why we're getting this increased decomposition rates in the moss mats, which leads to fragmentation. And this was a bit of a surprise for us. But there's a very clear increase in saprotrophic fungi associated both with the moss and in the soil. Okay? So this is a real functional group shift in relation to nitrogen increases. So that's been one of our studies, which we've just submitted for publication. That study was really the first large-scale study of alpine fungi in the UK, and it really showed some big surprises. And one of the most surprising one, perhaps, is this guy here, Squamanita contortipes. And this is a photo I was kindly lent from Jens. And this guy has been recorded once in the UK prior to this study. It was found as a single fruit body in Rothiemurchus, which is in the Scottish Highlands, in 1957. And that is it. That's the total records for this species in the UK. We found it in 8 of our 15 sites. And at one site, actually not so far from Rothiemurchus, it was in 5 out of our 8 samples from that site. So it seems to exist as a mycelium in the soil fairly commonly, but it doesn't seem to produce fruit bodies very regularly. And that's a real surprise. And there's lots of surprises like this. Another one, I'll just stay within the same genus. We have another Squamanita species from multiple sites, which molecularly is very close to Odorata. Again, picture from Jans. But it's not exactly Odorata. And we don't know what this is yet. So there might be another Squamanita species out there which is undescribed. And these are the kinds of records and finds that are going to turn up all the time when we go below ground and start looking at the cryptic diversity of mountain fungal communities. And it's great finding things like this. One other one which I'll mention is this guy here, Bankera violescens. And we have 3 sites for this, all near Aberdeen, in fact, and all under spruce. This, the one record that we have from the Alpine study below ground is right up, way up on the northwest of Scotland, from one of the highest mountains up there, totally out of its normal ecology. So again, I'm sure there's going to be lots of finds like this, and I think it's going to really force us to rethink the ecologies of some of these fungal species. So that was the first big study that we did down through the UK. We then decided to do a much more in-depth study, and we chose one mountain in the Cairngorms. It's called Ben Ann, and this is a fantastic plateau. It's the largest contiguous area of upland in the UK. It's about 5 square kilometres, and the whole top of it is covered by a fantastic mosaic of plant species, of plant communities. So the whole area is split up into well-defined zones, and those zones are determined, or the vegetation is determined by the topography. So the wind, snow lie, temperature, etc. And they're very, very clear patterns of vegetation here. And we wanted to know if the above ground heterogeneity in the vegetation was also reflected below ground in the biodiversity and the functioning of that biodiversity. This is the team which started all this. There's Andrea on the left there, who I've mentioned. We had an Italian student working with us, Ludovica Odi, and then Josie van Passen, Dutch lass. So this was the original team that set all this up. And you can see we're all in shirt sleeves, a nice day in Scotland. But a lot of the time it was much more like this. And this was one of the sampling trips up on the hill. And you can get these rather freak events where you can get quite a fall of snow. So it was rather an interesting experience. Just to show you what some of the vegetation looks like, this is Nardus grassland, and there's large areas of this incredibly tough grass up on the top. There's alpine heath, ericaceous heath, usually dominated by Calluna vulgaris, and those are the two of the big vegetation types, along with the Racemitrium heath. But the other habitat that we really wanted to include was the late snowbeds. And these are scattered across the mountaintop, and they are basically an endangered habitat because some of the climate change scenarios, which suggest we're getting a decrease in snowfall, and that within the next 50 years, we may lose most, if not all, of our late snowbeds in Scotland. And we really wanted to see how unique these habitats are before we actually lose them. So this, just to show you what a late snowbed looks like, in case you don't know, two of our plots are underneath this snowpack, and this is in May in 2019, and they usually become free somewhere the late June, perhaps into July, and then they have 3 months of snow-free time. And they're incredibly rich. They're very rich in bryophytes. They have snowbed bryophytes. They're very rich in myxomycetes, for example. And there are very few vascular plants that can cope with these conditions. The little one here, Nathalium, it's fairly common in these systems. And there's also a few annual grasses that grow in these late snowbeds. This just shows one of the plots that we had with some of the soil probes that we put in. So a very important, unique habitat. So what we did was we used national vegetation maps, and we set up 99 plots across the plateau, covering 7 different vegetation types. And 4 of us did about 24,000 meters of ascent in 2 weeks in July 2018. And we set up a huge diversity of plots, and we sampled all sorts of things. This is just to give you some idea of what we sampled. We had above ground, below ground, we had nutrient probes in the soil, collecting soil data. We dug soil pits to look at carbon and nitrogen stocks. We did decomposition studies. We had temperature measurements. It was a real comprehensive study, shall we say. And it was great fun setting it up. For the biology below ground, we collected samples, and we used what's called a metabarcoding approach. And we looked at bacteria, fungi, and then we also looked at all eukaryotes, that's anything with a nucleus, so all the animals, etc. And then we generated all sorts of sequence data from this approach. So we really tried to cover as much as we can, having gone to all the effort of setting up all these plots. So I'm just going to give you a real snapshot of some of the results that we have from this study. And there's one thing to consider, because even though we have done all this work and we've looked at all the soil biodiversity, all of the information that we have about the diversity comes from only 250 milligrams of soil, because that's what we could extract at that point. 250 milligrams from each plot. So in total, this is only 25 grams of soil from the entire mountain. And that's really worth bearing in mind. So what we have for the fungi, I'll just show you the fungi and skip the rest. So we managed to get a total of about 5.5 million DNA sequences from the study. For each of those little quarter-meter plots, we've got about 57,000 sequences. And in total, we ended up with somewhere in the region of 3,400 fungal species from the mountain. Okay? And within each of the little plots, we had over 200 species. So from this one study, from that mountain, we have over 3,400 species of fungi. Okay? That's about 3,400 more than what we knew before. Okay? So it's a giant leap forward with respect to knowing what fungi are up there on this particular mountain. Just to give you a wee bit of the data, this is the average number of species per vegetation type. So we've got snowbed on the left here, and then Juncus trividis, Vaccinium, the nardus, the grassland, the Racemitrium is the moss, and then Calluna and subalpine heaths. So there's a real gradient here of richness with respect to the fungi associated with each of these vegetation types. Okay? And you can see that the snowbed on the left really seems to have considerable diversity. And it really is. Whatever group we've looked at, the snowbeds contain unique assemblages of species. Just to show you how well we've picked out the communities, this is what's called an ordination of the fungal communities associated with the different vegetation types. And an ordination groups together communities which are most similar. So the points on here are individual communities, and the points which are close together means that those communities are very similar. And I can group them for you to show you a bit easier. So this group over on the left are the snowbed communities, the Juncus trividis, and the moss heath. Okay, so they seem to form a little group over there. We've got all the ericaceous heath, so that's the Vaccinium, subalpine, and then the Calluna. Not surprisingly, they all group together. And then the last one is the only vegetation type which is dominated by graminoids, grasses, and they form a little group down here. And this kind of strong association between above ground and below ground is really a feature of these communities. And I think these are communities, or these systems are fantastic to work in because they haven't been impacted yet. This is an area which is really devoid of man's influence so far. There's been a slight increase in this area with nitrogen deposition, but these are very, very strong interactions we're seeing between above ground and below ground. And that's something which is really quite unusual to find now. And it's one of the beauties of working in these systems. They are basically pristine systems, and you get these very, very strong interactions, direct interactions between above and below ground. And the fungal communities illustrate this beautifully. So that's the Benan mosaics. And like I said, the above and below ground communities and the functions, because we found links between the litter from the plants which are going in and the activities of the fungi in the soil. So there's really, like I said, there's very, very strong links between above and below ground, both in terms of their composition, but also in relation to their functions. And it's great to find these systems. The last study which I want to mention is an ongoing study, and this is an image of part of the Cairngorm range. And the Cairngorm National Park is the largest national park in the UK. It's about 1,100 square kilometres. To put that in context, it's bigger than Luxembourg. And it's a fantastic area which takes up or occupies a large proportion of the Central Highlands in Scotland. And it's a fantastic place for a holiday if you're looking for somewhere to go. So within the national park, there are 58 mountains over 3,000 feet. That's just over 900 meters. That might seem fairly low to you, but for us, 3,000 feet is a big mountain. And they have a special name in the UK. They're called Munros. And people spend a lot of their time going up Munros. They're called Munro baggers. And in Scotland as a whole, there are 282 mountains over 3,000 feet. So the Cairngorm National Park contains quite a proportion of those. And we thought it would be a great idea to actually sample the soil and the communities from all of them. So this is what we have been doing over the last year. And we teamed up with a charity called Plant Life. They have a network of volunteers who do things for them. And so we teamed up with Plant Life, and we asked volunteers to adopt a single mountain and then to sample the soil under 3 vegetation types. And we chose fairly distinct vegetation types. We gave them all the instructions. We sent them all the kit that they needed for sampling. And we got them to sample under the grassland, under the heath, and under racemitrium. And we also sent them a spare sampling tube, and we asked them to choose soil from whatever they wanted to know what was in it. And that was interesting. So we asked them to do all this over the last summer, and they were really great. We ended up with samples from 55 out of the 58 mountains, which is a fantastic return. And this kind of project is what's called citizen science project. I don't know if people know this term, but it's becoming very common in the UK to involve volunteers from the general public in science projects, which needs a lot of observations or samples collected over a large area. Apologies for the phone. So we were very successful in getting the samples back from the 55 mountains. And we have processed all the samples, and we are analyzing the data. And it's very exciting to see what's coming out of this. And what we know at the moment is the close association that we saw on top of Ben Ann, that single mountain, is also upheld across all the 55 mountains over the whole of the park. Okay, so we've gone from one mountain to 55 over this huge area, and this close association between above and below ground in specific vegetation types is upheld across all of these mountains in the park. So it's really exactly what we were trying to test, and to see whether or not these communities are superbly linked above and below ground. So we're really, really pleased at how this came out. So just to end up, we've gone from having almost no records and very little knowledge about our alpine fungi to really opening up the lid about our knowledge on their diversity and about their ecology. And they are very, very rich, species rich, and very functionally diverse. And they contain a lot of species that are either absent or rare, very rare, elsewhere in the UK. So, take home message: fungi are absolutely integral components of the biodiversity in our mountains. And every time you're walking across there, under each footstep that you take, there's probably hundreds of species. Okay? So just remember that next time you're out in the mountains. You might not see them, but they're there. Thank you very much.
Thank you so much for your presentation. It's another really interesting talk from you. Siri wonders how come the Scottish alpine fungal community was so poorly known?
That's a very good question. Most of the records came from nearby car parks, shall we say. We didn't have so many energetic mycologists, would be the simplest answer to that, I think. There were very few records.
I see. Eddie is wondering if the snowbird species diversity includes Navicolus myxomycetes or only true fungi?
It's what I presented was only the true fungi. We have the myxos in the other data set that we have. We actually have data on all 7 kingdoms from this study, which is quite amazing.
Yeah, that's really, really nice. Håvard is asking, the novel group Archaeorhizomycetes are very common in alpine areas. Common. Yeah. Have you looked into those?
Yeah, we have. Not specifically, because the approach which we use tends to underestimate them, but we certainly pick them up very commonly. We know that we are underestimating them because of the approach that we've taken, but we wanted to get as broad a view as possible.
No, I see. Do you know what the cause for this nitrogen pollution is?
Yeah, sure. Most of it comes from, in the UK at least, urban sources, agricultural sources, traffic and industry. So, and the effect is extreme. The gradient that we looked at, the Welsh sites are so degraded. I mean, we were putting in small litter bags that are only, a few centimeters in size, and I struggled sometimes to find patches of moss, which in Scotland cover hectares. I struggled to find pieces of moss that I could put them under to look at decomposition. It has just, they've lost it basically. And they used to have the same as what we had. It's really devastating.
Yeah. You also had quite a bit of cryptic species, such as this Squaminita. Is there an idea to look for fruiting bodies after extreme years with, for example, really hot weather or cold or dry or something like that?
Yeah, I would love. I've never seen one. No. And we see very few fruit bodies up there. Yeah, I would love to see a Squaminita. I don't count them as being cryptic because we can identify them. There's a huge component of the diversity which is cryptic, which we don't have any name for. We can only identify maybe 35% to species. Between 20 and 30%, you can only identify to fungi at the moment. That's how cryptic that is. Right? Yeah. The databases that we use as reference are still very, very underpopulated with reference material, unfortunately.
Yeah. So Håvard has a follow-up question in terms of this nitrogen issue. Yeah. In Netherlands, the nitrogen deposition has gone down and the ectomycorrhizal fungi recovered to some degree. Has this trend been observed in Scotland?
Fortunately, we don't suffer anywhere near the same as the Netherlands. I mean, in many areas in the Netherlands, you were getting levels of over 300 kilograms of nitrogen, which were devastating areas. That was the intensive agriculture. Those emissions have dropped dramatically. Although we have looked for impacts of nitrogen upon ectomycorrhizal fungi, the gradient within Scotland is quite short. And we've got some suggestions that we can pick up a nitrogen effect, But over the whole of the UK, the problem is that the climate, the habitats vary enormously across along the UK. So differentiating between an N effect and a habitat effect and a host effect is very difficult. I mean, we know nitrogen has a huge impact on ectos, that's established, but our gradients in Scotland are quite short. So you'd be difficult to pick up an effect and difficult to pick up a recovery.
Yeah, I see. Have you compared your study towards similar studies in other countries?
Yeah, there are not so many similar studies. There's individual studies looking at perhaps individual species. There's a few studies looking more broadly, but yeah, it's still very much an infant field. It's something which is gaining ground rapidly, but at the moment there's not so many studies. Most studies have so far come from Central European Alps, from Norway. There's a lot more from the Arctic, but we're dealing with different systems from real Arctic systems. So there's still a lot of work to be done.
Yeah, I'm sure. Okay, that was all the questions. Thank you so much for taking time and presenting your results to us.
My pleasure.
Okay, bye-bye.
Okay, bye now. Thank you.